Hello ladies and gentlemen, my name is Calvin, and today I will be presenting my research thesis titled Experimental Study of the Effect of Microbes' Existence in Marine Environment on the Corrosion Rate of Stainless Steel 304. This research was conducted under the expert guidance of my supervisors, Professor Dr. Tjokorda Tirta Nindhia and Professor Dr. Dewa Ngakan Ketut Putra Negara, and will be examined by three lecturers, which are Professor Dr. Insinyur Gusti Murarai Nindhia Santiarsa, STMT, Dr. Insinyur Iwayan Bandapatnyana, and Dr. Ibu Tulakantara STMT. I would like to start with the background of this study. Corrosion is a critical issue in the maritime industry, capable of causing both short-term and long-term structural problems. While stainless steel 304 is known for its corrosion resistance due to its chromium and nickel content, the marine environment introduces a complex variable: microorganisms. The presence of microbes leads to the formation of biofilms, which alter the interaction between the metal surface and the environment. This study was initiated to understand whether these microorganisms act as accelerators or inhibitors for corrosion on these specific materials. Based on this background, this research addresses two key problems. First, how does the presence of microorganisms impact the corrosion rate of stainless steel 304 immersed in seawater? Second, what are the specific differences in the corrosion forms created with and without microbial involvement? To ensure focused results, I established specific boundaries for this study. We measured the corrosion rate using weight loss method, recording mass changes at the end of every period. Our discussions focuses on the effect of microorganisms in general, without deeply analyzing specific microbial taxonomy. Finally, we utilize microscopic observation to compare the physical forms of corrosion between biotic and abiotic environments. The significance of this research lies in two areas. Academically, it enhances our understanding of the biocorrosion phenomenon and the specific impact of biofilms on stainless steel 304. Practically, it provides vital data for the maritime and structural engineering industries to develop more effective corrosion prevention methods, such as selecting better suited materials or coatings for biologically active environment. Let's briefly review the mechanism. Corrosion is the chemical decomposition of metal. In this context, it involves anodic reactions that ionize the metal and cathodic reactions that reduce chemical compounds, degrading the metal into its oxide forms. You can see the basic chemical reaction displayed in the image below, which is sourced from Wang et al. And Sarin et al. Studies. One common form of corrosion is uniform corrosion, occurs evenly across the entire metal surface. However, stainless steel 304 generally resists this well, due to its strong chromium passive layer. A more dangerous form for stainless steel 304 is pitting corrosion. This occurs rapidly and forms deep localized pits on the surface. It occurs when the protective passive layer is breached, often by aggressive ions like chloride found in seawater. Crucially, we also have biocorrosion, or microbiologically influenced corrosion. This corrosion initiated or accelerated by microorganisms. Their metabolic activities can damage the metal's protective layer and facilitate oxidation reactions. This figure contrasts the rapid mechanisms of microbially influenced corrosion on the left against the slower abiotic processes on the right side of the image. On the biotic side, bacteria accelerate corrosion through two pathways, which is EMIC shown by pathway A, where bacteria attach on the surface and strip electrons directly from the metal, and CMIC shown by pathways B and C, where bacteria excrete corrosive hydrogen sulfides that chemically attacks the iron. Conversely, the abiotic sites show standard chemical corrosion. Without the biological catalyst, the reaction is significantly slower than the biotic sites. Central to biocorrosion is the biofilm. This is a layer of microorganisms embedded in an extracellular polymeric synthesis matrix adhering to the metal. Wave films can trap high concentrations of corrosive agents and alter the electrochemical conditions at the metal surface, acting as either a trigger or an inhibitor for corrosion. This is a video recorded in the process of verifying the presence of microbial activity in the freshly taken seawater. The vibrating things in the middle is the microorganisms or bacteria that we want present in our freshly taken seawater. This diagram shows the overall steps that I did in my research, starting by preparing the material and equipment needed, weighing initial mass of each specimen, then immersing in both freshly taken seawater and sterilized seawater. But before immersing the specimen in freshly taken seawater, the presence of microorganisms was verified by observing a sample of the seawater taken with phase contrast microscope. After every period ends, which is 1 month all the way to 5 months, the specimens immersed were taken out of the water and cleaned by brushing. After ensuring the specimen has been thoroughly cleaned, it is weighed and its microstructure was observed. The weight data then used to calculate the corrosion rate of the periods measured. After the last period group of specimen data has been collected, the data was analyzed and used to conclude this study. Moving to the methodology, this study utilized a quantitative experimental approach. The research was conducted at the Manufacturing Industry and Machinery Research Group Laboratory and used seawater collected from the Jimbaran Beach. The process involves specimen preparation, initial weighing, immersion, and final analysis. Our procedure begins by weighing 30 specimens of stainless steel 3/4 with high precision scale. We created two immersion environments, which is fresh seawater that was replaced every 2 or 3 days to maintain live microbial activity in the water, and sterilized seawater, which is boiled to eliminate the microbes in the water. After immersion, we cleaned the specimens and weighed them again to determine mass loss. We then calculated the corrosion rate in 3 different units: grams per month, percentage weight loss per month, and mils per year. Finally, we observed the surface microstructure using a metallurgical microscope. The variables used in this experiment were divided into 3 types, which is the independent variables that include the presence of microorganisms, dependent variables which include the corrosion rate and surface corrosion from muscle samples, and control variables, which is the material itself and the environmental conditions. To ensure the consistency of the environmental conditions, we immerse and place the specimens in one place. We utilize standard precision equipment including digital scales, metallurgical microscopes for surface analysis, and laser cutting machine to ensure precise specimen dimensions. The other apparatus were container, clamp, kettle to boil hot water, PVC pipes, and glass base to mount the resin, tweezers, hair dryer, sliding caliper to measure the dimensions, and brush for cleaning purpose. The primary materials used were stainless steel 3/4 plates, Jimbaran seawater, resin for mounting specimens, and polishing materials like sandpaper and velvet fabric for microscopic preparation. This research timeline spanned 5 months, from October 2025 to February 2026, allowing us to capture the temporal evolutions of the corrosion process. To quantify our results, we used the standard ASTM-G1 formula to calculate the corrosion rate. We focus on mils per year, which integrates weight loss, density, surface area, and time. This standard allows us to predict the remaining life of a component. We applied the appropriate K-factor constants as per ASTM G1 standards to ensure accurate conversion between units. This table serves as a standard industry benchmark for classifying corrosion resistance, translating raw data into qualitative performance ratings. On the far left, materials are ranked from outstanding to unacceptable. These rankings are defined by the corrosion rate, shown primarily in mils per year. In the second column, with the equivalent metric units like millimeters per year provided on the right for conversion. Because stainless steel is a higher value alloy than cast iron, a rate in the fair, which is 20 to 50 mils per year range, might actually be considered too high for stainless steel. The table used before and the formula used to calculating the mils per year were taken from this book titled Corrosion Engineering, written by Part G. Fontana in 1969. This table presents our monthly data. A critical finding emerged here. The corrosion rate in the biotic environments fluctuated. It was high initially but decreased later. In contrast, the sterile environment showed a steady increase in corrosion rate, eventually surpassing the biotic rate in the final month. This phenomenon is known as a crossover. This is the data in the table shown as a graph. As we can see, in the 4 months, the phenomenon called crossover was happened. This is the same data served in the comparative way to easily see the difference of each period of time. Now let's visualize these results through morphology. In the unsterilized seawater, the surface showed a patchy attack. This patchy attack. We observe irregular deposits identified as iron sulfides, resulting from the activity of sulfate-reducing bacteria. However, as the biofilm matured, it became thick and acted as a barrier, inhibiting oxygen and slowing down corrosion in later stages. In the sterilized seawater, the surface remained relatively clean, with original rolling marks still visible. However, the damage here was structural. Without biofilm protection, the surface suffered from a stable pitting corrosion driven by chloride ions. These pits became deep and autocatalytic, leading to severe material loss by the 5th month. This is the same image plotted in as surface plot with the help of ImageJ software. This is the same image that is colored. On the right of each image, you can see the scale to easily identify the data from the color from the image. To conclude, the presence of microorganisms causes the corrosion rate to fluctuate. It is highly initially due to bacterial activity, but decreased later due to inhibitory effect of the mature biofilm. The sterile environment eventually becomes more destructive in the long term due to the accumulation of stable pitting corrosion caused by chloride ions. This is the crossover phenomenon. Morphologically, biotic environments result in uneven, patchy corrosion with iron sulfide deposits, while sterile environments result in deep localized pitting. For future research, I suggest extending the research duration to verify if the high corrosion trend in sterile environments continues permanently, and conducting molecular biological tests to identify the specific materials species involved in the biofilm. And this concludes my presentation. Thank you for your attention.